Welcome to Viral Illnesses and Antivirals, Part 1. We're going to discuss viral behavior and give you the basic understanding for the pharmacologic targets. So viral illness is the most common cause of human affliction and disease. And in my career, and now in the course of your career, you're going to come in contact more and more with these infections that we had not heard of before. Starting in 1983, HIV, and the upside of that was we came up with universal precautions by the CDC. There's also this term floating around called zoonosis or zoonotic infections, and that has to do with infections that seem to be based in the animal or, avian sources. So the SARS coronavirus is what gave birth to the severe acute respiratory syndrome, and that seemed to originate from bats, and you see the date here. And then you had MERS, which seemed to kind of somehow migrate from camels. We had Ebola and Zika, and you can see the case numbers there, which now brings us to the COVID-19 or coronavirus. The really key aspects of viral illness are things like how is it transmitted, who is colonized with it, what's the incubation period, where are the vectors, how is it spread, and what is the spread, the virulence of it. And you can learn more on this in Chapter 10 of McCants. The thing we know is that all viruses have a target cell, which is called the host cell. Here's the scoop on viruses. They have no organelles and thus are not a cell. They are simply a shell with an internal strand of nucleic material. So you've got the means to reproduce, and you've got this nucleic material that codes for certain proteins that the virus needs. It's protected by a shell. However, the virus's job is to bind to its target cell and take over. Viruses are classified in the three ways that you see, which is number one, by the type of nucleic material. Is it an RNA or a DNA virus? Secondly, they're classified further, is it double-stranded or single-stranded DNA or RNA? And you see the abbreviations here. Thirdly, do they use reverse transcriptase, which is an enzyme that is really significant for replication. And the main pathogen that we have right now is the HIV, which is a reverse transcriptase or a retrovirus. So the viral life cycle, because it's not living on its own, it has to bind to the receptor, invade, and take over. And it is in the taking over that it destroys the human host cell and it becomes a factory for itself. This then leads to destruction and spread. And you can see that the coding, or rather the classification of some of our most common viruses here. The adenovirus, which is your basic respiratory pharyngitis, or the herpes virus, is a double-stranded DNA. Coronavirus, single-stranded RNA. HIV is single-stranded RNA plus reverse transcriptase. See Table 10.6 in McCants for more. I need to talk a little bit about the human immune response, because when the human immune response does its thing, we kind of don't always need the pharmacology. But because it doesn't always do its things, because viruses are smart, just like bacteria, it's important to understand what should happen, what can happen, but then we'll talk later about the pharmacology. So the smaller the genome of the virus, so like the less complicated, the more rapid the reproduction and spread. And they live in our cells. So once they fuse and get into the cell, they can avoid phagocytosis and also avoid detection, because our immune system can only attack what it knows is there. So this is why there can be a little bit of a lag time with the immune response. Now, as you remember, the inflammatory response is our initial response. It's generalized, it's generic, it is not specific, and that's where you get the typical symptoms. So when our immune system does detect it, we get this cytokine storm, which is our interferons and our interleukins. Now, as you're going to see, viruses learn to evade interferon. Our secondary or our adaptive response, which is specific, takes 4 to 5 days, and that's when we get our T helper and our B cells for our plasma cells. Now, what has made HIV so incendiary is that its target cell is the CD4 T helper cell, which makes persons infected with HIV and AIDS susceptible to infection and malignancy because it kind of takes out our first part of our adaptive immune response. COVID-19, its target cell is the type 2 alveolar cells in our pneumocytes. Those are the cells that make surfactant, contributing to our elasticity. And surfactant also has some of a viral killing type of properties as well. The thing that we know about COVID is that it binds to the angiotensin II receptor, or ACE2, which is found in the lungs and in the GI tract. This slide on binding basics really can be a virus, it can be a drug. It's a ligand, which is a signaling molecule, plus receptor. The receptor is the receiving molecule, and they match. And every ligand has, not a universal, it just has a few little target receptors that it binds to. So then if you look, this kind of goes counterclockwise. So then over on the left, you got the HIV virus, which is from McCants, figure 10.9. And what you see is that kind of green thing, that's the internal capsid of the virus. And then you see those two little DNA strands. And then you see certain enzymes that the HIV virus has, integrase, protease, and reverse transcriptors. Those are all going to be pharmacologic targets, so just kind of study that for a little bit. I put this slide in here because it gives you the basics for understanding the pharmacologic targets. So what you see up on the top is your typical virus that has to fuse. It first must fuse to its target cell, and then it has to get inside the cell, get into the DNA and start transcribing. And so you can see it kind of takes over the cell, reproducing and going then out of the cell and finding other target cells. What you see on the left is the spike of the COVID-19 virus, and it is those are those little kind of sun rays that you've seen in all the pictures. Those spikes are what allows the COVID virus to bind, and remember its target is the ACE2 receptor, which is in the GI and in the lung. I also think now that there might be some fecal oral transmission because of the GI tract. That's kind of new and emerging. We're not 100% sure. We don't really call it viral resistance, but there are ways that the virus learns to evade our immune response. If you remember, we have MHC molecules on all of our cells. So one thing our body does is when the virus gets inside of a cell, the body kind of switches up its own identifier, its MHC, and that little red dot that you see there where I've got the arrow, the body, when it is infected by a virus, it adds a little piece of the virus, kind of signaling the cytotoxic T cell, hey, there's a live thing in here taking over me, come and help. So then the cytotoxic C releases cytokines to try to destroy the cell and stop the process. Then the next one shows, remember those little upside down Ys? That's our antibody, which comes from plasma cells. So this is showing virus particles that have been kind of absorbed and stopped by antibodies. And then the one on the bottom left actually shows phagocytosis, where the antibody plus virus particle have been absorbed and is going to get killed. So those are things that kind of happen. And that actually, the patient is symptomatic when all this is going on. This is also from McCants figure 1010, and I want you to study this. You can pause the recording or even go to your book. But this shows really some key things about HIV, which will help you understand the pharmacology, which will be in our next little presentation there. But you see where it's fusing and binding to the CD4 receptor, and then it kind of releases its material inside of the cell. And remember, there's some key enzymes that allow this to happen. So you've got reverse transcriptase, which remember allows HIV to kind of get into our nucleus and actually insert the RNA into our DNA, and it changes it to a double-stranded DNA. So it has to integrate into our nucleus. So it uses its own enzyme integrase. So we have a class of drug called integrase inhibitors. And then the next thing you see is the transcription happening and then it getting released out into the body. But just study this because there are classes of drugs for most of these steps. What we have here is a written out version of the previous slide of the steps to viral replication, which begins with attachment, and then we have fusion and uncoating, in which the RNA enzymes are released, the integration that I spoke about, and this is involving the reverse transcriptase plus the integrase enzyme. Then we have actual replication of the viral genome via transcription and translation. And then the HIV virus is going to continue to code for more integrase, more protease, more reverse transcriptase. And then it is packaged and it is dispersed out of the cell. So this is our final slide in this presentation because we're going to move into talking about the pharmacology for HIV. So we're going to have a whole bunch of meds called antiretroviral agents. As you know, a retrovirus is an RNA virus that carries RT, which allows the DNA molecule of the host cell to be taken over and replicate. One of the big terms that we'll talk about is viral load, because that's kind of how we target our therapeutic response. And the big thing is that we focus on early intervention, post-exposure prophylaxis. And the other thing I want to comment on is that HIV hits our innate immune system, which is our dendrites and our macrophages, as well as our adaptive, which is our CD4 cells. And that's the end of this presentation. Thank you so much.